Lauren is at the Lafayette School of Public Affairs at the University of Madison. She holds a PhD in economics, but also a Master of Science in genetics. She is one of these scholars who are at the forefront of trying to integrate social science data sets with dynamic understanding of biology to address some really fundamental questions that we as social scientists are interested in. What are the both behavioral and possibly biological factors driving social disparities? What are some of the profound influences that shape aging in health at older ages when we take account for individual behavior, their endowment, but also possibly a dynamic understanding of biology, where we think of biology not something that's just determined at birth through genetics, but actually evolves through the life course as a result of being exposed to various environments. So her research, not surprisingly, has received lots of funding from National Institute of Aging, National Science Foundation, lots of other funding sources. Her work appears in a very broad range of journals. On one hand, Journal of Epigenetic, on the other hand, Social Science Journal, Economics Journal, and that indicates the breadth of the work she's engaged in. So we're excited to hear your talk today that's going to be on leveraging the genome to evaluate the impact of the social environment. Lauren, all yours.
Thank you. Thank you. Thank you so much. I'm so happy to be here today and in person. This is exciting. I'm going to give a broad overview of my work in the field. So I'm not going to go too much in depth with any one study, but please feel free to raise your hand and interrupt me at any point in time if you want to know more details about anything that I'm showing you today. All right, let's get started. So I'm going to give you a general overview. First, we'll talk about incorporating genomic data into social science research. So I'll talk about why I think social scientists should care about genetics, computational and methodological challenges, and then I'll give 3 studies of mine that are examples of research on gene-by-social environment interactions that I've done. In the second part of the talk, we'll go into epigenetics. So I'll talk about some frontiers in epigenetic analyses in the social sciences. We'll talk about the specific type of epigenetic modification, DNA methylation in particular, that we'll be looking at. Some promises and pitfalls of DNA methylation data. Then I'll talk about the epigenetic clocks, which have emerged as very promising biomarkers of aging. And then how I'm integrating epigenetic clocks into my research on social adversity and accelerated epigenetic aging. So first, why should we care? And I think this is a big question, and it's an important question. And I'm just going to give you a few different reasons why I think it's important. First, and this might seem obvious, but I think it bears mentioning, that DNA is a critical source of human diversity. And because it's a critical source of human diversity, if we can account for it in our models, it allows us to explain more of the variation in an outcome. This then just increases our statistical power to detect effects, and it reduces omitted variable bias. So that's just from a very kind of broad statistical point of view. But overall, if we can account more for genetic variation, we can better evaluate the degree to which the environment matters. And that's really what's crucial to me as a social scientist. The second is that heterogeneity is the norm, not the exception. So we know that results at the mean do not tell the whole story. So what we're seeing here is a genetic risk distribution for breast cancer. I'll talk more about what a polygenic score is. Here it's just standardized, so the mean is 0. The red line is breast cancer cases, and the dotted blue line is controls. And you can see at the mean, there's no difference between cases and controls. So really what starts to get interesting in terms of looking at effects of the environment is if we look more at the tails, right? If we look beyond the mean. And this is where I think genetic data can really, really help us, because they can improve our understanding of heterogeneous responses to treatments or policy interventions. And I think they're an especially good variable to use to look at heterogeneous treatment effects, because they're an important source of human diversity, but also because they're determined randomly at baseline, conditional on your parents' genetics, and upstream from all other outcomes. So it's not itself an outcome of the social environment. And so because of that, I think it could be potentially a more useful variable to look at heterogeneous treatment effects than parental education, or something that itself is an outcome of the social environment and is therefore endogenous. The other is that genetic data are now available, molecular genetic data are now available in studies with really rich social measures. And this is because of just the massive decline in the price of sequencing the human genome. So in the over 20 years since the Human Genome Project commenced, since we had the first complete sequence of the human genome, the cost per raw megabase of DNA sequence, and 1 megabase is, I think, around 1 million base pairs of the genome, has fallen from $10,000 in 2001 to like a penny. And this was in 2015, so this stops, but it would just keep going like this. So it's becoming cheaper to genotype people than it is to collect survey data information on them. And because of this, it's emerging in studies like the Health and Retirement Study, the Ad Health Study, studies that we use that are the bread and butter of a lot of the work that we do. And it's not stopping anytime soon. So this really has allowed us to expand beyond twin and family studies in terms of thinking about genetics and actually really start working with molecular genetic data. But with great data come great responsibility. And this is huge. This is really, really important that we talk about the responsible and ethical uses of genetic data. So how should genetic data be incorporated into social science research? And can we use genetic data to inform research on social inequality and social policy? I think these are really, really important questions. They're questions that I hope my work speaks to, that I am actively thinking about and engaging in. Yeah, I think it's important, especially given the fraught history of the eugenics movement, that social scientists be at the table, that we be there to collaborate with geneticists, population geneticists, et cetera, to think about the best ways to talk about the environment and genetics and how they're coming together. Because like I said, these data aren't going away. If anything, we're gonna see more and more of it. So there's a lot of challenges, however, with incorporating genetic data. The first is computational. So, we're talking about massive amounts of data, typically around 2.5 million genetic variants that we can profile for each individual, so huge data matrices. So we need data reduction techniques that can allow us to capture biological specificity while maximizing statistical power. And that's where the polygenic score has really come in, and I'll talk more about what a polygenic score is and how I've used it in my studies. The second is methodological, and that is separating gene-environment correlation from gene-environment interaction. Now, there's many methodological challenges, but this is one that I've really worked on in my research. So we'll talk about those two distinctions and why they're important. 3 is interpretation of these genetic predictors for complex traits. So once we have a polygenic score, what does that mean? What does a polygenic score for educational attainment mean? And how do we think about that in the context of our models? Importantly, genes are operating through the environment, especially for incredibly complex outcomes like educational attainment. And so because of that, they're not deterministic. So how do we talk about these scores? And 4, and this is a huge one, is lack of genetic data in non-European ancestry groups. And what this means is that we're just really underpowered right now to conduct genetic association studies in diverse ancestry samples. So most of the work I'm going to be presenting to you today is on European ancestry populations. That is not something that, to me, is comfortable as a social scientist, but it's where the field of science is right now in genetics. It's not forever. We will eventually genotype the entire world, I'm sure, and then we will have the power to construct genetic scores in every ancestral group. But right now it is a limitation that we're working with. Like I said, it's not a limitation that's going to be there forever, and there are many, many brilliant minds in science working on how to overcome this limitation. But right now it does mean that a lot of the work that's being done, not just in the social sciences, but also in the biomedical sciences, is in large part on European ancestry samples. And so many of the gains to precision medicine are going to be going to European ancestry individuals. So that's something to just think about as well in terms of, overall challenges. Yes.
Has there been any increase in genetic collecting data with COVID, the rollout of vaccines and surveillance in different countries around the world? Has there been efforts to do that to collect gene data from populations?
You know, in relation to COVID, I'm not entirely sure, but
yeah, I mean, just the mechanism. If you have that many people out doing vaccine, I'm just curious if people are out in the field trying to
collect more genetic data kind of to it. It's a good question. I think it's something that I'm sure people are trying. I know there have been a number of big studies that have been done to look for genetic contributions to, you know, how quickly people, how severe COVID cases are and things like that. So I think certainly they've been doing that. Whether or not that's kind of increased the diversity of genetic samples, I'm not sure. It's a good question. I'm mostly going to be speaking to these first two today in the work that I'll show you. Any other questions before I? Okay, so the first is computational. So first of all, how do we even measure genetic differences across individuals? So here is just a figure that's showing how our DNA is wrapped really tightly around chromosomes so that it can fit into the nucleus of a cell. It's been compared to fitting Mount Everest into a grain of rice. So we're talking about massive amounts of information that have to fit into a tiny cell nucleus. If we unwind the DNA, we can see and go further, we can see gene regions. And then if we really zoom in, we see that our DNA is made up of these 4 nucleotide base pairs. So guanine, cytosine, adenine, thymine. And what we're interested in when we look at the genetic data are these regions of the genome, of these nucleotide base pairings that differ across individuals. So this is called a single nucleotide polymorphism. This is a single base pair on the genome that varies across individuals. If we just look at the genetic data matrix, this is what we see. So I have like the chromosome that the SNP is on, its position. So this is kind of like its geographic location. The SNP number, so each SNP has a unique identifier. And then we see the allele that was on each strand that you inherited from your parents. So we have, however, data, like I said, on 2.5 million base pairs. So how are we going to put this all into our model? We need data reduction techniques. So this is where the polygenic score has emerged. What we do when we construct a polygenic score is we apply summary statistics from a genome-wide association study to the genetic data matrix to create a single scalar of genetic propensity. So this is a Manhattan plot from a GWAS. How many of you have seen a Manhattan plot before? A few of you? Okay, yeah. I know Scott's, yeah. So what this is showing, so on the y-axis, we have the significance of the association. So I think this particular GWAS was maybe on depressive symptoms. So here we have regressions of depressive symptoms on each of those SNPs. Now they're separate regressions because we can't throw them all in the same regression. So each of these little dots are p-values from that particular association. And then on the x-axis here, we have where on the chromosome it's located. So it's called a Manhattan plot because it looks like buildings, tall buildings, skyscrapers. And then this dotted line is showing the cutoff for multiple hypothesis testing. So the kind of genome-wide level of significance. So these GWAS are especially best used in the case of really complex, what we call polygenic outcomes. So these are outcomes that are influenced by many genes. So you can see in this Manhattan plot, this is a whole genome event. There's not like one little area that's contributing to something as broad as depressive symptoms, right? Which we would expect. It's a really complex outcome. So what we do when we construct a polygenic score then is it's just formally a weighted average across the number of SNPs of the number of reference alleles. So you could have 0, 1, or 2 reference alleles at each of those SNPs. And then you're going to multiply that by the beta weight from a GWAS. So you're using this data from a really large GWAS. Sometimes now up to 1 million people are in these GWAS. And then you're applying that to your own data out of sample. So you're taking the weights from this GWAS, applying it to those SNPs in your sample, summing it all up. So it's basically just a weighted aggregate score. All right. So now let's jump into some methodological challenges. And that, to me, is distinguishing RGE from G by E. So what is G by E? This was a study that was done in 1982 by Anand Gupta and Richard Lewontin, who are two famous population geneticists. And what they were looking at here is how the mean bristle length on Drosophila, so these little hairs, how they evolve as the temperature changes. And what they found was that the mean bristle length evolved with temperature, but also based on a person's a Drosophila's genotype. So these different slopes are different norm or reactions to the environment based on genotype. So formally, it's just different norms of reaction to the environment because of genetic variation. And this is something that occurs in all species on Earth. So obviously, it's really challenging to study gene-environment interactions in human populations because we can't easily randomize different genotypes into different social environments like we can with model organisms. And so because of this, it's often really difficult to distinguish between gene-environment correlation or the phenomenon where we're selecting into an environment because of our genes from gene-environment interaction, or where the environment is actually moderating the genetic response. So because of this, what I've developed in my work is the use of quasi-natural experiments, and I'm just kind of calling this the Drosophila of the social sciences, so the model that we use to study causality in the social sciences. Why is it important to distinguish between the two? Well, with G by E, we can actually intervene. You know, there's room for a policy response because we can change the environment, and because of that, we can change the outcome. We can change the reaction norm, right, to the outcome. Past research has used twin and family studies to separate these two, but how should we approach this in population-based studies of unrelated individuals, right? Now we have all this genetic data in population-based studies, people aren't related, how do we do this? So this is where I've argued in my work that we can use quasi-natural experiments to study G by E. And here the G is a polygenic score, and the E is a plausibly exogenous measure of the social environment. So now I'm going to show you 3 examples of studies where I've done just that. Any other questions? I'll just pause for a second. I'm getting kind of winded talking in the mask, and I'm also just really excited. All right, so the first study I want to share with you is G by E interaction research on the Vietnam draft lotteries. So the random nature of the draft lotteries have been used to identify the impact of military service on health, education, and earnings. This has been just a kind of social experiment that's gone on for decades in economics and sociology. And past research has estimated the local average treatment effect or the rate of conscription, but only at the mean. And at the mean, we don't really find huge effects of the draft lottery of being conscripted and drafted into military service. Especially in the long term, it seems like there were maybe some effects right after the draft, but in the long term, the effects have been pretty small, and there really haven't been many effects identified, especially with respect to health. So what we wanted to do in this study was look at, did the effect of military service on smoking behavior vary by genetic predisposition? We're going to use just a basic 2-stage least squares approach to estimate the G by E. Our sample is 631 European ancestry men born between 1948 and 1952. We're using the health and retirement study for this. Our E is military service instrumented with the draft lottery, and our G is a polygenic score for smoking behavior. So this figure on the right kind of just sums up our findings. It's showing the difference in predicted max cigarettes per day between veterans and non-veterans, again, as instrumented by the draft lottery. On the y-axis, we have predicted cigarettes per day, and on the x-axis, we have the smoking units of the smoking initiation genetic risk score. And it's standardized, so the mean is 0. And what you see here is at the mean, and this is what past studies have found, there's no difference between veterans and non-veterans in terms of how much they were smoking. And this always seemed a little bit improbable to me because especially with the draft lottery, when these soldiers were in the field, they were given free cigarettes in their rations. So they had tons of cigarettes to smoke, and I can imagine they did. I would have too. What a stressful life event. So, but in terms of the genetics, no difference at the mean. But what was interesting is that we did find differences when we looked at individuals with higher genetic risk. So there, individuals who were drafted and went into the war were more likely to smoke and become lifetime smokers than individuals who were not. Individuals who were not, who had the same genetic risk score. So here, this appears to be evidence of a gene-environment interaction.
Does that hold it? Do you control for parental smoking status here? Can you
We
control
don't,
that?
we can't. We can't. Oops, sorry. But, you know, that was upstream of the draft, so. But yeah, unfortunately, they don't.
And is this risk score, is it it's specific to actually smoking tobacco?
Correct.
Addiction?
Correct. No, this is a smoking initiation polygenic score. So it's specific for initiating smoking. We also looked at there's also a score that's specific for cigarettes, number of cigarettes smoked, and the results were similar there.
Have you done any comparisons to like temperament, like if someone's temperament towards addiction, compare that?
Oh, interesting. I have not.
You could say that genes are embedded within that, but I'm just curious, like if something is kind of clear measure.
Yeah, no, I have not, but that's a good question. Others, yes?
Can you say a little bit more about how this genetic risk score is developed? It's based on a different data set?
Different data set, correct.
So purely based on correlations between genes and behaviors on this separate data set?
Correct. And then so we're taking those outside predictions out of sample and applying them to our sample. Yes, any other questions? Yes?
Is the study limited to European American veterans, given that most of the genetic research is on Europeans?
Yes. So that was part of my dissertation work. So since then, I've gone on to look at other ones. This is GBI research on the US high school movement. Claudia Golden, Larry Katz, others have written about this incredible phenomenon that happened starting in the early 1900s, up through the 1960s into the 70s, which was this rapid growth of secondary schooling that happened in the first half of the 20th century, and it's been termed the high school movement. This rapid rise, these are Claudia Golden's words, not mine. She says it was nothing short of spectacular, and it was not happening anywhere else in the world. So the US was kind of the first to see this huge expansion in high school education. And what we wanted to ask in this study was whether these substantial expansions in high school completion, did this reduce genetic disparities in educational attainment? And here we're going to exploit variation in high school graduation rates across states and over time. So we're using like a traditional diff and diff, where we're exploiting differences in average high school across states, so based on the state you were born in and lived in when you were going to school. So our sample is over 8,000 European ancestry individuals who were born between 1930 and 1959. And our E here is high school completion rates by state of birth, sex, and 6-year birth cohort from the US Census. And our G is the educational attainment polygenic score. So here you can see just some basic regression results. The outcome here is GED, or high school completion. So you can see with just the high school completion, that state level measure that we constructed, that is highly associated with a person's own educational attainment. The polygenic score is also highly associated with high school completion. And then we have their interaction, which was highly significant and negative. And we'll talk a little bit in a second about what that negative interaction means. But this was pretty robust no matter what we threw in there. So we have year birth fixed effects, state fixed effects. We haven't gotten a chance to talk about genetic principal components, but those are on all of our models. Region times year birth fixed effects, all of that, additional state controls. And to kind of visualize what's actually going on here, I think we have to look at some marginal plots. So on the y-axis, we have the predicted probability of obtaining a GED or high school degree. And on the x-axis, we have the polygenic score, again, for educational attainment. Again, mean standardized, so 0 is the mean. This top line is a state with above average high school completion rates. So this is a state that's 1 standard deviation out in high school completion rates. The bottom line is a state with below average high school completion rates, so 1 standard deviation below. And what I find fascinating is that you can see there's no difference in high school completion by genetics, genetic risk score here, for individuals who are in high-performing states. So regardless of your underlying kind of genetic predisposition, you are just as likely to graduate from high school as someone with a really high genetic predisposition. Not so much in states with the low completion rates. So here we see that individuals, if you had a really kind of high genetic predisposition, you were more likely to graduate from high school than an individual in the bottom. So I think this is showing something really powerful about social inequality and about social policy. Although we're not pinpointing any particular policy here, we can imagine that states with higher high school graduation rates probably have policies in place to promote high school education and so forth, compared to the low-performing states. And we can see here that that does make a difference. There's inequalities that are emerging based on a person's underlying genetics, but that those inequalities don't have to be there, right? They don't have to be deterministic.
Just practicing on many today about polygenic score, but what does that actually mean to have a propensity to complete high school? Do they have, is it ability, cognition, things like that? Do they link that at all?
Yes, no, that's an excellent question. That's, it's something I haven't really talked about. And you kind of hear me struggling with this too as I say it, because I don't want to call it genetic endowment. I don't want to call it, it's hard to kind of pinpoint exactly what to call it. It's an underlying summation of a bunch of SNPs that have been found to be highly associated with educational attainment. They have been linked because they do functional studies in these GWAS to see, okay, great, we found this SNP. Is it linked to something that makes sense? Most of these SNPs are in regions of neurocognitive development. So they do seem to be playing a big role in cognition and in the development of neurons in the brain. So it does seem like the SNPs that they're identifying in these huge GWAS are in fact linked to neurodevelopment. Yeah, great question.
So maybe you're getting there, it seems to me that this kind of work that you could look at changes in state compulsory education laws. And then the other thing that
Yes.
within it, if you have a big enough sample, like in a state like Illinois that rates high, you can see that's just driven by like Chicago and the suburbs. If you're in downstate Illinois, I was thinking this was tassel corn in the summer. So I mean, you have, I wonder if you can look within a state to see if this gap changes, because you have like an urban area where everyone's going to high school, and you have a rural area where if you work on a farm, you might not have much of a sense.
Yeah, those are both great questions. So to the first question, sorry, what was your first question again? Compulsory schooling, thank you. I would have loved to use compulsory schooling laws. The problem is the HRS cohorts were born from the 1930s onward. By then, compulsory schooling had already kind of had its strong effects. I do have a colleague at another university who's looking, because now the HRS is linked to the census, the 1940 census, is looking actually at how compulsory schooling laws affected the grandparents and how that then, this kind of G/E with the kids. So that's, stay tuned. But I'd love to get more specific in terms of policies, but it's hard with these cohorts. And then, you're right, I should look at urban-rural differences. I haven't done that yet. Unfortunately, I can't get any more fine geographically than that, although now I could use the 1940 census. So, I need to think about that more. But right now, this is out at the state level. Okay. And the final G/E study I want to show you is on job loss and BMI. And here I'm going to use what's called a VPGS, or a variance polygenic score. And I'll explain what that means in just a minute. So, in the social sciences, the effects of job loss on health have been studied extensively. The scarring effects for older workers are particularly severe. But I would say results on a whole have been really mixed for weight gain and changes in weight as a result of job loss. Some studies have found evidence of weight gain in middle-aged workers, but overall there's no consensus. So what we wanted to ask in this study is, does genetic risk moderate changes in BMI after a job loss? Our E here is job loss from a business or plant closure, which in the research has been kind of pinpointed as being slightly more exogenous than layoffs or firings. So these are situations where everybody at the plant loses their job, not just a single individual. And we're going to use a difference in difference estimation with propensity score matching, again in the health and retirement study. So here the treatment group are workers aged 50 to 70 who report they are no longer working with their previous employer due to a business closure, and the control group is same-aged workers who worked for the same employer. And our G here, we're going to use both an MPGS, or basically a main effects PGS, which is what I've shown you so far. That's the polygenic score we're used to seeing. But then we're also going to incorporate something new, which is a VPGS, a variance polygenic score. So what is a VPGS? Why should we care? So with this standard PGS that I've been describing so far, it's based on a GWAS of main effects. So we're looking at the effects of each SNP on an outcome, and it's the average effect of that SNP on an outcome. But the question is that really an ideal measure for capturing variance in an outcome that's the result of the environment, right? We're just capturing main effects, but we're not really seeing the variance around that. So what a variance polygenic score does is it actually captures aggregate genetic effects on phenotypic variability. And so really what we think these are capturing is what's called a VQTL effect. That's a variance quantitative loci. Those are SNPs on the genome that actually affect the plasticity of a trait, so how fast an individual adjusts to their environment. There are new GWAS methods out. I'm highlighting one here by Alexander Young and colleagues that decouple SNP and variance effects to estimate these dispersion effects on phenotypic variance. So basically you're able to then estimate kind of the phenotypic variance independent of the general mean. So what we do is we use mean effect weights from an MGWAS to construct the MPGS, and then we use these dispersion effect weights from a VGWAS to construct the VPGS. So here's the G/E results for BMI with the MPGS and VPGS. Here our outcome is BMI controlling for BMI in time t-1. So you can see here at the top just the kind of basic main effects of business closure. We don't really see any effect. So that's what past studies have found, no effects of business closure on BMI. If we add the MPGS model to the model, we can see that it's highly predictive of the outcome. So this is a polygenic score that's been designed to be predictive of BMI, and we're seeing that it is indeed predictive of BMI. These are just FDR-adjusted p-values, by the way. Then we have its interaction. So we interact the MPGS with business closures. Nothing. We then put the VPGS into the model. Its main effect isn't significant, which we would expect, because we're not looking at the variance of BMI as the outcome. But its interaction is. So here it seems that the VPGS is doing a better job of capturing that phenotypic variability due to underlying genetic differences than the MPGS. If we show this graphically again, here you can see we have this is again results from the full model, and we're controlling for a ton of stuff here with the propensity score matching and all of that kind of stuff. You can see here just level effects with the MPGS interaction, nothing going on. Higher levels of BMI with higher MPGS, that's what we would expect. Here with the VPGS interaction, we can actually begin to see differences emerge between the treatment and control groups. This is kind of what we would expect to see that might be going on with the gene-environment interaction. So this was really interesting to us. I think this might be the first paper to use a VPGS in G/E research. So we were excited about this, and yeah, I'm excited to see more work coming down the pipeline on this. All right, so I'm going to pause for a second before I go into epigenetics. Any questions about G/E, polygenic scores? Yes? Yeah, no, that's a great question. So this generally means that people with lower variance polygenic scores are adjusting to their environment more slowly. So they're basically higher variance polygenic scores means you're more plastic. You adjust to your environment more quickly. With a lower polygenic score, you might. And this was really crucial in this study because we're only measuring BMI every 2 years. So I think this graph would look very different, or this figure, if we were able to measure BMI more proximal to the job loss. But so I think really what we're seeing here are these are people whose BMI just adjusts more slowly. So we can still see the effects of the shock 2 years later. That's my hypothesis. And what we found too in the HRS, again, these are in older individuals. BMI is not easy to deal with in older individuals because actually weight loss, it can be worse than weight gain. It can be a sign of disease and accelerated aging. So actually maintaining BMI and even somewhat gaining weight as you age can be a good thing. So even here, we're actually seeing weight loss was the outcome. And it wasn't until we were able we worked on this for a couple of years, and it wasn't until this technology came out with the VPGS that we were able to really capture this. So this is just an example of how the science is evolving so quickly that we can start to incorporate these techniques more and more to make sense of what we're seeing. Great question, thank you, because I forgot to mention that. Any other questions? Okay. All right. So how many of you have studied epigenetics at all, or are kind of just curious about epigenetics, or, no, it's intriguing. And I think as social scientists, we're especially intrigued by it because it's responsive to the environment. So the environment can affect our epigenome, and that's something that has captured, I think, the imagination of a lot of social scientists. So up until now, what we've been talking about is GWAS. We've been talking about this relationship between genetic variance and some sort of clinical trait, and this is measured now statistically with the GWAS. But we're ignoring all these really complex molecular processes that help take our genome, all these kind of variants in our genome to an actual clinical trait or outcome. So we're going to be talking now about the epigenome, which is this kind of regulatory element space. So formally what it's doing is it's actually regulating gene expression or transcription of genes across cell division cycles. And the type of epigenetic modification that we're going to talk about today is DNA methylation. This is the most studied type of epigenetic regulation, but it's not the only one. So that's just to let you know, DNA methylation is not the only type of epigenetics, but it's the one that's most commonly studied, and it's the one that is cheapest to assay right now. And formally, that's just when a methyl group gets added to a DNA molecule at the cytosine base. And it's a CPG site is a site that can get methylated on the genome. So this is literally just a kind of a methyl group is being added to the cytosine molecule. And you can see here, C always pairs with G. That's why we call it a CPG site. And typically, when these methyl groups attach to these CPG sites tend to be really overrepresented in gene promoter regions. So these are regions of the genome that are regulating gene transcription and kind of whether the actual machinery, transcriptional machinery can attach to the genome and then create an mRNA transcript, right? And that RNA transcript goes out into the cell, outside of the nucleus, into the ribosome, and it creates a protein. But in order for that transcription to even happen, we have this regulation by the epigenome. So when it's located in a gene promoter, it's usually repressing gene transcription. So it's kind of acting as a dimmer. It can also upregulate gene transcription, but basically it represses it usually. So when we look at DNA methylation data, we're no longer looking at these distinct categories of how many alleles you have at a given site, 0, 1, 2. We're looking at a continuous outcome. So we're looking at the percent of methylation at a CPG site. And again, as I mentioned, DNA methylation is changeable over your life course, and it's responsive to the environment. It is also influenced by genetics, but it is also influenced by the environment. So there's a number of different things that one could study with epigenetics, but what I'm gonna show you today is this research on epigenetic clocks. And these have emerged as really promising biomarkers of aging. So an epigenetic clock is just a composite score of CPG sites that are highly associated with chronological age or mortality risk. And as it turns out, the epigenome, as we age, it actually exhibits really precise transformations with age. So it's something that we can see the aging process in the methylome. So formally, it's just built by regressing chronological age or phenotypic age on CPGs using a supervised machine learning method. So it's just a weighted linear average of methylation levels. So here we're using like, enet or other machine learning methods. And that machine learning method is picking CPG sites that are really highly associated with some outcomes. So it's here usually trained on chronological age or phenotypic age. And then you can take the betas, again, the beta weights from that outside study and apply it to your study to calculate an individual's epigenetic age or biological, really their epigenetic age. But it's a measure of biological aging. Yes? Say that again?
How do you come up with the supervision, like setting what the expectations are? It's unsupervised learning algorithm versus unsupervised.
Yeah, so I'm not exactly sure the different settings because I haven't myself constructed these clocks. I'm not sure the different settings that they use, but they're using ElastiNet most of the time. And I'm not sure what other boundaries they're placing on the model, but I think they're pretty minimal. So basically, we could take, if we took your blood today, we could put it into this algorithm, we could calculate your DNA methylation, put it into this algorithm, and it would tell me what your epigenetic age is. And then we can compare that to your chronological age to see if you're aging faster or slower than your chronological age. And so this is the outcome that we're really interested in, which is epigenetic age acceleration. This has been going on for a decade, this research, and it's highly associated with age-related diseases and mortality. It is more predictive of mortality than other measures of biological age, than any other phenotypes of aging that have come out so far. So if you're interested in aging, this is important. I don't have a lot of time to go into this, but so basically there's a number of different clocks that have been developed. And overall, they show very little overlap across CPG sites. And I think this is really because they're constructed using different training phenotypes, different training samples, and across different tissue types. So the clocks first kind of were developed back in 2013 by Steve Horvath and Genome. So these initial clocks, I'm calling them first generation clocks, these were really just predictive of chronological age. They were designed to be predictive of chronological age. This second generation clocks, they decided we really actually want to have these be more predictive of mortality and mortality risk. So these were trained more on phenotypic aging and then actually on time to death. So they're much more predictive of mortality. And then the third generation clocks that have been developed by Dan Belsky are actually, instead of comparing CPG sites across individuals, it's looking at individuals of the same chronological age as they age over time. So he calls these pace of aging measures, which is slightly different from the clocks. So I received a K99R00 award from the NIA to do this research on linking social determinants with epigenetic aging clocks. And what we wanted to see here was, do epigenetic clocks capture information about exposure to socioeconomic environments and related biological pathways of aging? The studies that had come before this showed inconsistent results. They were mostly using the older clocks. Here we wanted to use some of these newer clocks, and we wanted to use two large aging studies that now have epigenetic data, MESA and the HRS. We looked at various dimensions of socioeconomic status and how they were related to 8 different epigenetic clocks in 2 well-powered population representative samples. And then we also looked at whether these associations were mediated by smoking, BMI, alcohol consumption. So this is just kind of the summary slide of what we found. Here we have 8 different clocks, those clocks I showed you before. Here they're ordered kind of in order of when they were created chronologically. These are all standardized effect sizes, so you can actually compare the effect sizes across these different clocks. Model 1 is the more saturated color. That's just the SES index that we created. The clock progressed on that with just age, sex, race, and some technical covariates. Here we are able to use diverse ancestry groups. We didn't get to touch on that, but it's not as big of an issue with epigenetics. In Model 2, then we add these lifestyle behaviors. And what we found here were that the newer clocks, the Levine, the Yang, the Grimage, the Dunedin Poem, were much more predictive, or much more, I shouldn't say predictive, socioeconomic status was more likely to be associated with these newer clocks. And again, I think it's because these newer clocks are actually capturing something more proximal that's related to mortality and time to death. So here we saw, the worse you were socioeconomically, the faster you were aging epigenetically. And finally, I have one more study to show you. I'm excited about this study because it's looking at specifically in utero shocks and epigenetic aging. So in general, I would say it's really challenging to study the causal effects of early life shocks on epigenetic modifications. The Dutch hunger winter studies famously did this. That was like really groundbreaking work. That was causal. But overall, I would there's really a need for research that can identify sensitive or critical periods when the environment has its greatest impact on epigenetic aging signatures. So what we did with this study was we wanted to ask, does exposure to economic fluctuations in utero impact late life epigenetic aging? Can we see, something that happened to you when you were in utero, can we still see patterns in your epigenetic aging signature decades later? And what we're going to do is focus on the most severe and prolonged economic downturn in American history, the Great Depression. So why focus on the Great Depression? It was a massive economic shock. A quarter of the US labor force was unemployed, fortunes were destroyed, and importantly, there was no social safety net at the time. And there were no prenatal vitamins. There was no maternal care. It was 4 to 5 times the size of the Great Recession, et cetera. Importantly, also, there were really dramatic geographic and temporal variation in economic conditions surrounding the Great Depression. And this was really due to kind of differences in manufacturing versus agriculture in the different states, and also in subsequent relief from the government through the New Deal. So what we're going to do is exploit state and year level variation in macroeconomic conditions during the 1930s to identify the impact of the Great Depression on epigenetic aging. And we're going to use the health and retirement study again. But what's really exciting about the HRS, and if you all and students especially are interested in working with epigenetics, this is a phenomenal opportunity. This is the first nationally representative study with epigenetic data that has the geographic variation necessary to actually assess kind of if where you were born matters for how you age. So it was especially exciting for me to be able to use these data. This is the wage index by state. So this is the exposure variable that we're using, which is just wages in the state that you were born in. These are farm and non-farm wages. So you can see, these are indexed to the year 100, 1929, before the Depression. Wages fell dramatically, and then, during the New Deal, there was a lot of, obviously, kind of help that came in from the federal government. Things recovered. But here, just to note that there is a lot of variation across states and also across birth year in terms of what people were exposed to. We use just a basic difference in difference event time study specification. So here, the outcome again is epigenetic age acceleration measured for individual I, born in state S and region R, et cetera. Their blood was taken in 2016. So these are people who were born between 1930 and 1940. Their blood was taken in 2016. The wages are our exposure here. So this is just the aggregate wage index at the state and year of birth in the period before conception. So we have a pre-trend in utero and then in early childhood. And we go all the way up to age 16. Number of controls, fixed effects for identification, et cetera. Happy to talk more about that. So this is just, before we look at the event time study results, this is just the regression of the wage index in utero on the outcomes. And you can see here we don't find anything again for the earlier clocks, but the more recent clocks, again, we do find highly significant effects. These were very robust across different model specifications and controls. So basically, we found that a 1 standard deviation increase in the wage index in utero led to about a 0.3 standard deviation decline in GRIM age, epigenetic age acceleration, and POAM, epigenetic age acceleration, years later. This is very similar to what others have found in the early childhood literature. So example, the effect of food stamps and so forth on metabolic syndrome in adults, very similar effect sizes there. And just to give you a sense of the magnitude, a 1 standard deviation of the wage index is equivalent to just about 80% of the overall decline in wages that happened during the Great Depression. This, however, is the figure that I get really excited about. Yes?
Go ahead, talk about this one, then I'll ask that question.
This is the full event time study specification. Here this is from the fully specified model. So we have, the individual, for each individual, we have the wages, the year before they were born, their wages that they were exposed to in utero, and then the wages that they were exposed to all the way up through age 16. So this is an incredibly stringent model. And it's all in utero. So this was exciting because, of the work that's been done on fetal origins and fetal programming. And here, too, we're seeing this persist even in individuals in the HRS who survived to be actually, have their blood drawn in 2016. So yes, mortality selection is like the elephant in the room. If anything, it's just biasing our effects downward. The fact that we could still see something even amongst those that survived. So here, people who had, higher wages in utero are aging slower epigenetically than those that didn't. So there was some sort of maybe mechanism of programming that was going on there. This is the same, we see the exact same event time study kind of landscape when we look at the Dunedin POAM, epigenetic age acceleration. So those were those two that seem to be really capturing what we're seeing. So just general takeaways here, we see economic fluctuations from the Great Depression, increased epigenetic age acceleration among individuals who survived until 2016. The effects of the shock appear to be linked to the in utero period. And we looked really extensively, I didn't have time to get into it today, at selective responses. So we looked at mortality, we looked at fertility around the time. We don't find those to be driving our results. We also don't find that our results are driven by the Dust Bowl states, by New Deal spending, or by World War II. And I'm happy to talk about any of those things in more detail. So what's next in social epigenomics? This is my last slide. But I want to just talk about some exciting things that I think are coming down the pipeline. Yes?
I just want to say you have a few minutes.
Yes, I'm watching the time. As I mentioned in epigenetics, these study populations are really important in how we're constructing these clocks because each study population is undergoing different environmental resources or constraints, and that seems really key to illuminating perhaps some of these different social pathways of aging. So what I'm excited about and work with Hans-Peter Kohler and with Scott Damrauer, who's also in the room, and Ileana Kohler, is that we're looking to collect epigenetic and genetic data in the Malawi longitudinal study of families and health. So here we really want to see whether clocks constructed in high-income countries are capturing aging in the same way that these clocks might be that are constructed in low-income countries. So we'll actually be able to really look at epigenetic aging in a very resource-constrained environment in Malawi. I think there's also a huge need for causal work in social epigenomics. So this is, we need more natural experiment work. We need more randomized control trials, et cetera, whatever. This is really important. There's basically no causal work in epigenetics other than the Dutch hunger winter, the Quebec ice storm study. Those are the two that come to mind. So I have some other work that's looking at the causal impacts of smoking behavior on epigenetic signatures using variation in cigarette taxation. So that's something else that I'm excited about. And then finally, we can also use the epigenome to improve how we construct polygenic scores. So this is something, we can use all this annotation to actually see, to construct tissue-specific polygenic scores. So the epigenome, it's regulating gene transcription, but it's also regulating cellular fate. So whether a cell becomes a muscle cell or a skin cell or a blood cell, that's something that the epigenome is regulating. So we could use those methylome patterns to readjust SNP weights from a GWAS to then construct tissue-specific scores. So you could construct a tissue-specific addiction score for the brain, for example, using this, these epigenetic data. So tons of data coming down the pipeline, lots of exciting stuff, especially for graduate students who are into biosocial work. So I hope this provided you with a nice overview. I want to say thank you to my research assistants, my collaborations. This work is not done in isolation. It is team science work, and I'm grateful to a number of collaborators for all of their insights, to my research assistants, and also a number of funding acknowledgments from the NIA, the Russell Sage Foundation, March of Dimes, National Science Foundation, the NBR Retirement and Disability Research Consortium, and also the Center for Retirement Research through the Social Security Administration. So thank you. Follow me on Twitter. Didn't used to have to say that at the end of my talks, but now I am. Thank you so much for your active participation. Appreciate it. Thank you.